Welcome to Cancer Research UK RadNet Manchester. Based at the University of Manchester and working in collaboration with the Christie NHS Foundation Trust, we provide world-leading radiotherapy research across a range of technologies and treatment areas. Our aim is to improve survival and quality of life for all our patients. Today, we'll be discussing some of our work investigating interactions between immune cells and radiotherapy.
Radiotherapy is delivered to about 60% of cancer patients, and while care is taken to specifically target the cancerous sites, a boundary of normal tissues will inevitably be in the radiation field. This may lead to side effects, which limit the quality of life of cancer survivors, some of which are acute, but some may progress to long-term severe side effects, such as chronic inflammation, fibrosis, and organ dysfunction. With the aim of understanding the normal tissue responses to radiotherapy, our collaborative project has a specific focus on the immune responses to radiotherapy within the mucosal barriers. We are investigating how localized radiation modifies immune cell populations within the irradiated intestine, the draining lymph nodes, and systemically within the entire body. Our hypothesis is that the initial irradiation triggers a cascade of immune changes which involve the endothelial glycocalyx, epithelial cells, and the released cytokines, chemokines, danger-associated molecular patterns, and growth factors including TGF-beta. This then primes the niche and recruits immune cells to the site of radiation injury. Some of these immune responses are beneficial for cancer control, and some are helping with tissue recovery. However, the questions we are asking are, what is changing in this irradiated niche that initiates and sustains the inflammation and injury that cannot recover over time? What are the key immune players? How do they interact? And how are they involved in these processes?
Following radiotherapy, locally produced chemokines diffuse and are transported to the endothelium, facilitating immune cell recruitment. For immune cells to leave the circulation towards the tissues, they must pass through the endothelial glycocalyx barrier. This is a thick layer that lines the vascular endothelium and inhibits leukocyte recruitment. However, it is poorly understood how radiotherapy alters the chemokine production, the glycocalyx structure, and how this regulates intestinal inflammation and fibrosis. Having developed a mouse model of fibrosis, we can now explore if the glycocalyx components are shed after radiotherapy and how this contributes to the overall immune cell recruitment, affected niche alterations, and the long-term side effects. To address these important questions, we use novel murine models including knockout and reporters of inflammatory chemokine receptors and activators of TGF-beta. We will also investigate whether the use of specific inhibitors prevents glycocalyx degradation and thus leukocyte recruitment. These new insights will provide an enhanced understanding of the mechanisms driving the radiation-induced side effects. Identification of potential therapeutic targets that can alleviate the radiation-induced toxicity can benefit patients' recovery following treatment.